Everyone, let me discuss about enzyme linked immunosorbent assay. Uh, Interaction. The interaction between antigen and antibodies. Uh, the reactions are highly specific and antigen react only with antibodies produced by itself. So antigen antibody interaction means that a specific antigen can bind with a specific antibody. So, like that, antigen antibody reactions. So that the antigen antibody reaction will be detected. Yeah, mm, will be detected by many process. So now we are going to discuss about only the Elisa technique. So they have been used in a many diagnostic tests for the detection of either the antigen or the antibody in vitro techniques. So you know the very well about the in vitro. So in vitro, usually the test will be performed under the laboratory condition, especially in glass vase. So this is like a in vitro test. Antigens and antibodies bind through non-covalent bonds in a manner. Protein proteins binds to their cellular receptor or enzymes bind to their substrate. The physiochemical properties of this antigen antibody reaction. So the electrostatic bonds, hydrogen bonding, van der Waals bonds, and hydrophobic interactions are the intermolecular forces involved in antigen antibody reaction. Affinity denote the intensity of attraction between antigen antibody complex. Low affinity antibodies binds antigen weakly and tend to dissociate readily. In case of high affinity reactions, more tightly for longer duration. So low affinity interaction. So then that antigen antibody complex may be dissociate within a short period. But the high affinity, if the antigen antibody binds with the high affinity, so that is also stable for longer duration. Then avidity is a manner of the overall strength of binding of an antigen with many antigenic determinants and multivalent antibodies. Antigen Antigen antibody reaction will be measured by different types of techniques. So then now we are going to discuss about the enzyme linked immunosorbent assay. Enzymes conjugated with an antibody react with colorless substrate to generate a color reaction product. So antigen antibodies, so then both of them are known as colorless substances. So the uh, colorless substances we are not able to find out during the antigen antibody complex formation. So that's why they are also using some technique. The enzymes will be conjugated with antibodies. Then after the substrate bind with enzymes that will produce specific color reaction. So that color reactions will be measured with the help of a spectrophotometer. The substrate is called a chromogenic substrate. A number of enzymes have been employed for ELISA, including uh, alkaline phosphatase, horseradish peroxidase, and galactosidase. Following these are also known as the enzymes. The enzymes will be used to uh, conjugate. The enzymes will be used to conjugate with the antibody molecules. This assay approach the sensitivity of RIA, radioimmunoassays, and have the advantage of being safer and less costly. ELISA allowing qualitative detection or quantitative measurement of either antigen or antibody Each type of ELISA can be used qualitatively to detect the presence of antibody or antigen. Then a standard curve based on known concentrations of antibody or antigen is prepared, from which the no unknown concentration of a sample can be determined. So, in ELISA techniques, usually they have to follow. They have to follow. Unknown sample will be uh, unknown sample will be uh, compared with standard, the known standard curve. So known standard, then already they have to collect values, the values against the known sample. Then after, the unknown sample value will be compared with known standard value. Indirect ELISA, so the antibody can be detected or quantitatively determined with an indirect ELISA. Serum containing primary antibody, or like the patient serum. So the patient serum also containing antibody is known as primary antibody. So that primary antibody is added to an antigen coded microtiter well and allowed to react with the antigen attached to the well. The presence of antibodies bound to the antigen is detected by adding an enzyme conjugated secondary anti-isotype antibody. Then finally, the amount of color reaction product that forms is measured by specialized spectrophotometric plate readers, so which can measure the absorbance of all the wells of 96 well plate within seconds. So indirect is nothing but the direct reaction. So the antigen antibody binding reaction, also known as direct reaction. Here with indirect ELISA, so initially they have to collect the patient serum. So the patient serum contain primary antibodies against disease. So then the suspected antigen. So the suspected antigen previously loaded on the microtiter plate or ELISA plate. Then after, the antibody, the patient serum containing antibody will be added on the same antigen coded well. Here with the developed a specific antigen antibody reaction. Then finally they have to add the enzyme conjugated antibody which was specific for the primary antibody or human antibody. So the secondary antibody, also known as the specific antibody for a human antibody. So that antibody can bind with human immunoglobulin. So then after, you have to add the substrate. So the substrate bind with the specific enzymes. Then after, color will be developed. The color formation will be detected with the help of a spectrophotometric reader. Indirect ELISA is the method of choice to detect the presence of serum antibodies against human immunodeficiency virus (HIV). In this assay, the recombinant envelope and core proteins of HIV are absorbed as solid phase antigens to microtiter wells. Individuals infected with HIV will produce serum antibodies to epitopes on these viral proteins. The second, another one type of ELISA, sandwich ELISA. So antigen can be detected or measured by a sandwich ELISA. In this technique, the antibody is immobilized on a microtiter well. So you know very well that a sandwich. So sandwich, how they were prepare the sandwich. So initially they have to prepare someone bread. Then after, they have to add stuff, nutritive material. Then again they have to place another one bread. So this structure also like bread, stuff, again bread. So the same like this ELISA preparation. So initially we have to fix immunoglobulin. So immunoglobulin on the ELISA titer plate well. Then after that how to add antigen. A sample containing antigen is added and allowed to react with immobilized antibody. After the well is washed, a second enzyme linked immunoglobulins specific for a different epitopes on the antigen is added and allowed to react with the bound antigen. After incubation period, after the completion of incubation period, so the free second antibody, that means unbound antibody, is removed by washing process. Then finally we have to add substrate. If the will contain the enzyme conjugated antibody, so the substrate can easily bind with enzymes and forms color reaction. So the intensity of color will be measured by the spectrophotometric reader. Then after the results, the test sample results will be compared with known standard results. Then another one type of ELISA, competitive ELISA. Another variation for measuring amount of antigen is competitive manner. In this technique, antibody is first incubated in a solution with a sample containing antigen. The antigen antibody mixture is then added to an antigen coded microtiter well. The more antigen present in the sample, the less free antibody will be available to bind to the antigen codal well. Addition of an enzyme conjugated secondary antibody specific for isotypes of the primary antibody can be used to determine the amount of primary antibody bound to the well as in an indirect ELISA. In the competitive assay, however, the higher the concentration of an antigen in the original sample, the lower of the absorbance. So in a competitive assay manner, the higher concentration of antigen in the original sample, the lower the absorbance. So the image also depicts all these three types of ELISA: direct ELISA, indirect ELISA, then a competitive of first one, indirect ELISA. Initially they have to fix antigen and the antigen in the ELISA plate well. Then after they have to add patient serum containing antibodies. So here with developed antigen and antibody reactions. Then in a third well they have to add enzyme conjugated secondary antibody specific for primary antibody. So then secondary antibody binds with primary antibody. Then after completion of the incubation period, so we want to wash and remove unbound antibodies from the reaction well. Then finally we have to add substrate. So the substrate can bind with specific enzyme and produce color reactions. So that color intensity will be measured by the help of photometer. Then second methodology is sandwich methodology. So initially the antibody is also fixed in the ELISA plate containing well. Then after incubation, so then we want to wash out unbound antibodies from the well. Then again we have to add specific antigen. So antigen, here it occurs specific antigen antibody reaction. Unbound antibodies unbound antibodies or antigens are removed by the washing process. Then finally we have to add the specific antigen which was conjugated with enzyme. So the specific or primary enzymes are added on the same well. So if the antigen specific for antibodies, enzyme conjugated antibodies, so the antibody can easily bind with antigen molecule. This reaction will be occur sandwich. Initially immunoglobulin, then after antigen, then again immunoglobulin. So this reaction will be a pure sandwich model. So then finally we have to add substrate, then substrate specifically bind with enzyme and produce color reaction. So that color intensity will be detected with the help of spectrophotometer. Third one, competitive manner. So competitive manner, then antigen antibody pre-incubated. So antigen antibody pre-incubated, then after the pre-incubated antigen antibody complex transfer into the Elisa plate well. The Elisa plate will also contain antigen. So then antibody here it's occur the competition between the antigen and antibody. So then next after completion of the incubation period, so we want to remove the floated antigen antibodies. Then finally you have to add enzyme conjugated antibody and substrate. Then color intensity will be detected with the help of spectrophotometer. Thank you. Thank you one and all for watching this video.